Dr. Omar, I think now you can start.
السلام عليكم جميعا الأساتذة والحضور الكرام. إن شاء الله اليوم إنه موعد جديد مع ورشة عمل مع ضيف آخر دكتور سليم اللي يتكلم لنا الآن من جامعة ساسكاتشوان PhD candidate. أهلا وسهلا بك دكتور سليم.
أهلا.
Dr. Slim, just a small, like a simple introduction. Dr. Slim, he has finished his DVM in 2012 from the National Veterinary Medicine School of Tunisia, and he's finished a master's degree in biomedical science from Texas A&M University in 2017, and he is doing PhD, and he has PhD candidate. Inshallah, he will graduate. Next year, right?
Yeah,
He'll graduate.
Yeah, correct.
And his PhD in vaccinology immunotherapeutics at the University of Saskatchewan, Canada. So you are welcome, Dr. Slim, today with us.
And yeah, thank you very much, Dr. Omar, for the invitation, and the College of Veterinary Medicine in Baghdad for this invitation to present my previous work for my master thesis at S19. So basically the title of this presentation is vaccine safety study for Brucella, abortus, S19 and S19 delta VGBR in pregnant swine. And I chose this title simply because it's the title of my paper published in 2019. And as you can see here, like Dr. Omar was part of the research and most of the experiments. He gave a tremendous help toward the realization of this work. If you're looking for a lengthy version of this work and an extended version of this work with more experiments and some parts, like a second part of my work, you can go to my thesis titled Towards the Development of an Improved Vaccine for Swine Against Brucellosis. So you can find it online. It's visible for everyone. So without further ado, I'm gonna start with an outline. So my presentation will start with an introduction to brucellosis. Then I will go to the goals and objectives of the study, my experimental design and results, and then I will finish with some conclusions and acknowledgements, of course. So what is brucellosis? So brucellosis, also called multi-fever or ambulant fever, is a neglected zoonotic disease caused by Brucella species. It's a gram-negative intracellular bacteria, nearly has a worldwide distribution. It causes 500,000 human cases per year, and it has a huge economic impact due to the loss of livestock through abortion. So mainly the disease causes abortion in pregnant animals. So this table will summarize the preferential hosts of the different pathogens, different species of Brucella. So I'll start with Brucella abortus, which has as a preferential host the cattle. Also, you can find some infection with Brucella militensis, some rare Brucella suis infections. In my study, I focused mainly on the swine brucellosis and the causative agent of swine brucellosis is Brucella suis. But you can find some rare cases like due to Brucella abortus and militensis. And most of them, they are coming from majorly spillover from wild animals to the domestic animals. So in the state of Texas, it's a huge burden coming from the feral swine, and I will go in some details later on. So the diseases in human is described by flu-like symptoms, indolent fever, of course, body aches, but then we'll have severe complications as osteoarticular infection, gastrointestinal infection, lymphadenopathy, hepatobiliary and hepatomegaly, genitourinary, so majorly orchitis in males, and it induces abortion in females. We can see also some complication, cardiovascular complication and neurological complications. In animals, as I said, we mainly see abortions in females. So it's described through hemorrhagic cotyledons in the placenta. We can see also abortion in the pig, as you can see in this picture. In males, we can see epididymitis in the testicles and hygroma as an articulation inflammation. The transmission to humans is mainly through ingestion of unpasteurized the dairy products. There's also direct inoculation through cuts and skin abrasion while through contact with close contact with animals. So mainly we see it like in the veterinary medicine through handling of animals, and also without wearing gloves, usually without wearing gloves when handling abortion products. Through also animal carcasses, as well as placentas, and contact with animal vaginal secretion without protection. Also, it can be through aerosol, so keep in mind 10 to 100 CFU can cause infection in humans. The transmission to animal is due through ingestion or contact with infected tissues or placenta, or also body fluid, as you can see here in the picture. So that's the placenta after either abortion or giving normal birth through contact. So animals tend to eat the placenta or the placenta products from other animals. It can also be due through venereal transmission in swine. We can see that mainly in sheep and dogs. There's also, as I said, spillover from wild animals. Usually we see it from the feral swine to the domestic animal. In the state of Texas, so brucellosis has confirmed, as you can see in red, has confirmed infections majorly in feral swine. So the state of Texas is Brucella-free in domestic animals, but it's not free in the feral swine. So it represents a danger through the spillover from the wild animals to the domestic ones. The control and the eradication of brucellosis. So there are three major strategies that have been demonstrated as effective tools to control brucellosis in domestic animals when used in combination. So usually we start with strict biosecurity at the farm level. So building fences, no contact with other animals. Like if you raise animals like let's say cattle, there's no contact with sheep or no contact with swine, and so on. So we do also screening and slaughter programs. So screening if animals like are positive, we proceed directly to killing. So that's how we kind of do a sanitation of the entire herd. And also vaccination of the susceptible population. So vaccination, there are different type of vaccines that I'm gonna talk about later on. So the different vaccines, we have killed vaccines like the Brucella abortus strain 4520. It's a heat-killed vaccine. It's not used anymore. Subunit vaccines. These are kind of like promising strategies, but not existing. So people are trying to do research to develop subunit vaccines because they are less receptors, but they are poorly immunogenic. So basically, what we do is we add adjuvants to the subunit to the basically immunogenic antigens. We add adjuvants. DNA vaccines also, multiple antigens can be expressed, and possibility of co-expression of multiple cytokines as adjuvants. But mainly here, the ones that are basically industrialized, and you can find them in the commercial. We can find the live attenuated vaccines. So for cattle, I don't know about Iraq, but in Tunisia, we still use the Brucella abortus strain 19. In cattle, in the US, they don't use it anymore, so they use the Brucella abortus strain RB51. simply because you can distinguish between infected and vaccinated animals. Brucella abortus strain 19, you cannot distinguish between infected and vaccinated animals. So that's why the policy in the US changed toward of the Brucella abortus strain RB51. For Brucella militensis, there is the strain Rev1 in sheep and goat. For Brucella suis, so let me tell you since the very beginning, there's no commercialized vaccine. The only country in the world that has a commercial vaccine and used the vaccine is China. That's only existing in China, and it's delivered through the drinking water of animals. So I'll let you imagine how biohazardous it is through the canalization of water, and it's not appropriate to use it honestly. So, and on top of that, it induces abortion if administered intramuscularly. So we're not preventing basically the abortion. So how those are kind of like the things that made us decide to go and try to develop a vaccine and try. So basically, there's no commercially available vaccine for swine, and Brucella abortus S19 has been used in the US for swine brucellosis, but in some kind of like trial. They did not persist in the usage of the S19 because of its safety and some mitigating studies. I'm talking about the 50 here. So some mitigating results were saying that they have seen abortion after vaccination, but at the same time, those studies, they were basically not only vaccinating animals through an endemic herd. So we didn't know if the abortion is due to the vaccine itself or it's due to the infection. Using the RB51, still, these are two vaccines for cattle, but in trial, we are trying them in swine. So usage of the RB51 is non-protective against Brucella.
Yeah. العفو آسف على المقاطعة. Just the sound. The sound is not clear for all of us. I mean.
Okay, can you hear me right now?
Yes, and just like be slow a little bit, please.
Okay, sounds good. Okay.
I mean, you are using the headphone?
I'm using my headphones, but let me try to take them.
Be close, please.
Let me try to take them off and see.
Now is it clear? Now it's very nice.
Hello?
Say it again.
Hello, hello.
Ahlan.
Can you hear me? Can you hear me? Okay.
Yes, it's very nice now. Go ahead, please.
Sound good. So coming back to the studies made on pigs using RB51, so they were not protective at all against the Brucella suis challenge in a study in 2006. Okay, so our strategy in our lab was to develop a live attenuated vaccine called the Delta VJBR as a promising vaccine candidate for brucellosis. So in Brucella, there's something called VRP operon, which is kind of like a virulence factor of the type 4 secretion system. So it's a major virulence factor of the type 4 secretion system. I have a picture here that shows the type 4 secretion system constituted by the different VRP genes. And most of them are very important to the translocation of virulence factors to the cell membrane, So here we have the bacteria, we have the inner membrane, the outer membrane of the bacteria, and then we have the cell membrane of any cell, majorly like the macrophage or the trophoblast. So the substrate will go from here and will be translocated to the cell membrane. So these are substrate virulence factor substrates. So the VJBR has been known that it controls and regulates the expression of the entire operon, but also it contributes to the survival and replication of Brucella. So it's very important. So earlier study in our lab in the mice demonstrated that the deletion of the VJBR gene will induce protection against challenge in mice and will reduce Brucella colonization in the spleen. So, and we don't see any splenomegaly or hepatomegaly. Another technology used in our lab is through encapsulation, so microencapsulation. So it basically enhances the efficacy of the delta VJBR. So basically the vaccine strain, the live attenuated vaccine, is engulfed within an erodible alginate microcapsule containing non-immunogenic protein designated VPB. And basically the vaccine will be inside this empty capsule. And one of its good effect, it sustains release of the vaccine strain over 36 days. That has been proven in the mouse model. Okay, now let me go to our goals and objectives of the study. So our long-term goal was the development of a safe and efficacious vaccine against Brucella species for animals and humans. Our short-term goal was to develop a safe and efficacious vaccine for the swine brucellosis using the microencapsulation as a controlled delivery system. So my study was limited to the safety. I did not proceed to the efficacy in my master study, so I did only the first part of it, which is the safety. So our first objective was to demonstrate the safety of the S19 delta VJBR vaccine candidate, and then to determine the humoral immunity induced by the vaccination. So these were my main two objectives. Now I'll go to the experimental design and show my results. Okay, so we had 15 American Yorkshire gilts. All of them were Brucella-free at the very beginning. We did the screening, and they are coming from a Brucella-free farm. We did they were artificially inseminated ahead, and they were acclimatized for seven days, and the pregnancy was confirmed by ultrasound after 60 days post-insemination. And all of the animals were microchipped for identification. This is the safety study design. So we vaccinated the pregnant gilts at mid-gestation, 60 days, through the subcutaneous route with a dose of 10 to 9 CFU per animal. Our different groups were Brucella abortus S19. We have 4 animals, so every group has 4 animals. Brucella abortus S19 delta VJBR encapsulated through the technology of the microencapsulation. Brucella abortus S19 delta VJBR unencapsulated, and we have empty capsule as a control group. And then we monitored majorly the animals for abortion, for adverse reaction, temperature fluctuation, and shedding of the vaccine. So the temperature, and we did also vaginal swabbing, and we did also blood screening, basically in 2, 4, and 6 weeks post-vaccination. And then we looked for abortion and normal farrowing, and we went and did bacteriology study and histology study, just to see if there are any inflammation or if there is any shedding or any infection. As I said, we monitored majorly all of the gilts for adverse side effects associated with vaccination. So first thing first was abortion, the most important thing. Other adverse reactions, body temperature fluctuation, and shedding of the vaccine through the screening of the vaginal swabs of the pregnant swine. This table will summarize the pregnancy and the delivery of the piglets. So basically, at the end of the study, we had 181 piglets. So that's the litter size. Each group has almost equal litter size. So we had 49 in the first group. Second group, the delta VJBR encapsulated, we had 49 piglets. The Delta VGBR unencapsulated, we had 45 piglets, and the control group, we had 38 piglets. We went right away to the abortion. Let's go right away to the abortion. So we have seen no abortion inside any group. So no abortion seen. Most of the things that we encountered was postpartum deaths. So we saw in the different animals like different number of piglets that died postpartum. So the cause of the death was majorly aggressiveness from the gills. Keep in mind these gills were primiparous, and that kind of like behavior was described normally. So all of these postpartum deaths were not due to the vaccination. We've seen stillbirths in one stillbirth in the first group, which is vaccinated with the S19, and also in the control group, just to say that it's normal. So stillbirth is encountered as well in primiparous gills. We've seen also in the first group a mummified animal, and in the control group, just to say, as mentioned, that this is normal. It's not due to vaccination or whatsoever. So in terms of following up with the body temperature, we have not seen through the course of the entire study any fever. So all of the animals, as you can see here, so fever is here noted by the dots. And as you can see, all of them, they were below the threshold. So no fever encountered. The bacteria in vaginal secretion, as I said, we did different vaginal swabbing for the gills at the pre-vaccination, and also 2 weeks, 4 weeks, and 6 weeks, and we have not encountered any. So basically what we do is that we take the vaginal swab and we culture it in TSA plates, and we do the follow-up and to see if there is any colony growing. And as you can see, none of the groups showed growth or bacteria. No bacteria colonization in the gill tissues after delivery. So we collected different tissues. So blood, liver, lung, spleen, mammary lymph nodes, inguinal lymph nodes, also the uterus and the placenta. Nothing. We have not found any bacterial growth. We complemented that with the H&E staining of the different tissues. So as you can see here in the slide, so we collected the spleen, the liver, the lung, and the uterus, and these are the different groups. And thankfully to Dr. Omar, who helped with the H&E staining and the interpretation of these results. So we did not find any inflammation or whatsoever in the different tissues. These are basically the different tissues of the gills. Then we proceeded to the piglet tissues. So we collected the liver, the lung, the spleen, the umbilicus, the kidney, and the stomach. None of them demonstrated any growth of bacteria. We went on and we collected different tissues as well. So for the umbilicus, the lung, the liver, and the spleen, these are, as I mentioned, different tissues from the piglet, from the different vaccinated gills. And we did not find any modification of the architecture of the different tissues. So in summary, vaccination of gills with S19 or S19 Delta VGBR did not induce abortion or fever. Following vaccination, no bacterial colonization was found in the vaginal secretion swab culture. So that our vaccine is environmental safe. Bacteria was absent in tissue homogenate culture from gills or piglets. So there's no vertical transmission of the vaccine strains. No significant changes in histological tissue architecture was observed following vaccination. So the S19 and S19 Delta VGBR vaccine strain seem to be safe in swine. Then we went to our second objective, which is the confirmation of the vaccination by serological tests. So first test, what we did is the Rose Bengal test. It's a rapid and sensitive qualitative test. It's basically an agglutination reaction seen if the sample is positive, and it's 1-to-1 Rose Bengal antigen and serum, so 30 microliters. To complement the qualitative test, we did a quantitative test, which is the indirect ELISA. So for that, we used Brucella abortus 2308 lysate as basically as an antigen, and we used the animals here at different time points through the collection, as I said, pre-vaccination, 2 weeks, 4 weeks, and 6 weeks post-vaccination. The first test is the Rose Bengal test. So in this picture, I'm showing the 4 positive. So these are kind of like annotation of the test. So we gave like a mark of 4 positive to the one that shows like high agglutination. Negative, as you can see here, we don't see any agglutination. And these are the different groups, as you can see here, and the different animals. So pre-vaccination, as mentioned, no animals had Brucella, so that's very comforting. At the second week post-vaccination, we started to see agglutination in the different groups. So as you can see, most of them were positive in the first group, vaccinated with S19, second group as well, third group as well. Only this animal, the 4th animal in the group vaccinated with S19 Delta VZBR encapsulated, did not show agglutination. And in our control group, no animals whatsoever showed because they were not vaccinated. They received only the empty capsule. Okay. Then we proceeded to the ELISA. And as it's kind of like correlating, so the results of the ELISA are correlating with the Rose Bengal test. So at the pre-vaccination, none of the animals were positive, and at the second week, we see a statistically significant rise of the IgM here. Similarly, we see a rise in the IgG as well, starting from the second week. Here is the control group. So they are all compared to the control group. And as you can see, they are statistically significant. Starting from the 4th week in the IgM, they start to decrease. Here, it's kind of like a progressive decrease. So, and at the 6th week, we start to see basically they have the same magnitude and they are not statistically significant. So basically, the peak OD values of the IgM and IgG were at 2 weeks post-vaccination. It entirely correlates with the previous results here, as you can see here, second week. All gilts were positive at second week post-vaccination. So even the one animal that was negative with the Rose Bengal was confirmed positive after 2 weeks. Negative IgM OD values at 6 weeks post-vaccination. Here I'm adding another, like this figure here is coming from a paper showing the durability of the vaccination with S19 in terms of total IgG. And as you can see, the drop of the total IgG starts at 210 days post-vaccination. Just to summarize, there's a transient immune response in the pig compared to the cattle. So in summary, vaccination of gilts with S19 and S19 Delta VZBR was confirmed by Rose Bengal test and indirect ELISA. As I mentioned, transient humoral response of swine as opposed to cattle. In conclusions, no introduction of abortion in swine following vaccination with, let me correct it here, it's 2-10-9 CFU of S19 Delta VZBR vaccine during mid-gestation. Both S19 and S19 Delta VZBR vaccine strain seem to be safe in swine as no environmental settings through vaginal secretion was detected. Transient humoral response in swine after vaccination, which was 6 weeks. So we saw the decrease after 6 weeks as compared to the cattle, 210 days. Possibility of using the Rose-Bengal test or indirect ELISA to distinguish vaccinated from infected animal. As I said, so infected animals in swine, so they show like kind of like a plateau of IgG. Whereas vaccinated animals, as I said, they start to show a decrease in 6 weeks. So basically, we can use the Rose-Bengal test or the indirect ELISA to distinguish between the vaccinated animals and the infected ones. Future studies is obviously the performance of an efficacy study. So vaccination of pregnant gilts with S19 or S19 delta VGBR, depending on the formulation, can be microencapsulated or unencapsulated, and then challenge them against Brucella fluid. Efficacy study will demonstrate whether or not the vaccine candidate is protected, as we don't know if that transient humoral response can be protective or not for the pigs. And with that, let me thank all my former lab members, starting with my PI, Dr. Arenas, Daniel Garcia, who helped a lot with the experiments, Dr. Omar Khalaf and Lance Wheeler. Here are our brucellosis team members, Dr. Thomas Fick, Dr. Alison Fick, and Dr. Shaki Sankar and Dr. Gary Adams. And I won't forget that I was sponsored entirely by the Fulbright scholarship to finish my master's. And thank you, and I'm open to any question.
Thank you, Dr. Steen, for this nice presentation and this amazing job you have done. And I think you forget to mention that you published this paper, right, in Vaccine?
Oh yeah, I put it like in the first slide, Omar. So I said, if you are curious to read the paper or the thesis, it's available online. So let me go back here. Yep, it's here. So that's the paper and that's the thesis, basically. There's an extended work that I did not talk about today in this presentation. Yeah. So basically, we tried to develop a DIVA diagnostic tool to distinguish vaccinated with the Delta VGBR from the infected animals. So that's the continuity of this study that you can find it in the thesis.
Thank you so much, Dr. Steen. Please, you can, if you have any question, you can raise your hand to ask Dr. Steen about his topic.
Yes, I see Dr. Dalia. Go ahead.
Hi, Dr. Slim.
Hello.
Thank you for your introduction. It was nice, but you was talking so in hurry. I don't know why. So
I'm sorry about that.
It's okay. So I have two questions actually. First one, so which kind of vaccine you used? Is it the DNA vaccine?
No.
Can you hear me?
Yes, I can hear you.
Okay, so was it a DNA vaccine or subunit vaccine or attenuated?
Yeah, so it was a live attenuated vaccine. All of them are live attenuated vaccines.
Okay, and can you tell us about the encapsulation?
So the encapsulation, honestly, I don't have like further knowledge about it, but I can go to the slide.
Okay, so is it a kind of lipid materials with it, and how you can deliver it? You know, so we have two materials, the vaccine and the capsule. Okay, so how you mix both before you deliver to the host?
Yeah, so basically,
Okay, then the mechanism of taking the capsule with the vaccine.
So there's a kind of like a system micro encapsulation device that has kind of like a nozzle. So basically, the entire mechanism is using the alginate, and also, as I remember, that was fasciola hepatica that was coming from fasciola hepatica eggshell, and it was proved like by our and patented by our lab that it will allow a slow release of the live attenuated
Please,
vaccine.
can you slow down?
Yeah.
So this capsule,
Yeah.
as I correct you saying that it's from fasciola hepatica?
Yes. Yeah.
Okay.
Yeah.
Okay. Yeah. Which part?
Fasciola hepatica eggshell.
Okay. Okay.
Yeah. Yeah. And also the alginate. So the alginate was used, but those are like, as I said, kind of like a glimpse for me because I did not produce the vaccine. I only did the trial. So, so the knowledge, my knowledge is kind of like a basic knowledge regarding the microencapsulation. As I know so far, and Dr. Omar can correct me or add to that, we used a microencapsulation machine that has a nozzle that will create, you know, the capsules, and in which we include the live attenuated vaccine. So the live attenuated vaccine will be inside the empty capsule, basically. And then that's it. We inject the liquid into the animals.
Okay.
Yeah.
Okay. Thank you.
Yeah.
Thank you for the answer.
I see several papers that published by Texas and AM about the eggshell precursor protein in a whole. So if you could refer to the first paper that described the composition or how they produce these capsules, that would be wonderful. Even if it's not now, later will can Dr. Omar distribute this paper to the audience, because I think they are interested in such thing.
Yeah, for sure. I can share with that. And actually, Dr. Alison Fick, the one that I acknowledge at the end, she's the one who developed this technique. So basically, all of her papers will describe like in details how she did, and I can also like send papers to Dr. Omar if interested.
Sure, thank you.
Yeah.
Thank you so much, Dr. Sarhad and Dr. Slim.
Yeah, Dr. Omar, you can give any feedback about the study, how hard it was.
Exactly, it's so hard for us. I mean, to follow up the result, because the professor, they think the VJBR like will be the best and S19, but the summary of this study showed, I mean, S19 and S19 delta VJBR, they have both like efficacy to protect like against the abortion. I mean, because they think about VJBR, and they had a lot of papers about VJBR, like in mice and pregnant mice. And after like Dr. Slim graduated, I mean, they followed with the with different studies, guinea pigs, pregnant guinea pigs. In general, the VJBR is safe. I mean, this vaccine is like safe and didn't induce any abortion, even like in swine, in guinea pigs. And they were trying to do like more models. And Dr. Slim, I remember like you talked, I mean, in our like previous like meetings, we wanted to do like double knockout. I think one time Dr. Tom Fick mentioned that.
I think he mentioned that briefly, but I don't think that they continued with the double knockout. So and I think the delta VJBR is fairly attenuated, honestly, because it disrupts the Verbi operon, but also it disrupts the VCEC. So the VCEC is the virulence factor, and the VJBR is kind of like a transcriptional regulator of the VCEC. And the VCEC is usually like incriminated in the abortion. or through the killing of the cells of the trophoblast. So that's why I think the Delta VGBR was a very promising vaccine, live attenuated vaccine, in comparison to the S19. Remember that the S19 induces abortion, whereas the Delta VGBR does not induce abortion in cattle or in mouse, or you see what I mean? So it's safe as a vaccine. Whether it's protective or not, we don't know. So we have to proceed to the efficacy studies in the different species, whether it's cattle, whether it's swine, whether I don't know, it's sheep, because I know that in my former lab they worked with militensis as well. So they worked with Brucella militensis Delta VGBR as well. So
I remember, like, after you graduate, they had a project in BL3 in Colorado State University in the vet school, and they had a study and using S19, and I think they have also VGBR from Brucella militensis, VGBR.
Yeah.
And both of these vaccines, I mean, they were safe, but sometimes they didn't like protect when they do like challenge.
When they do the challenge.
Exactly. I mean, this is the problem with this VGBR, like candidate. Is this like is safe, but it's not protecting. And they also conduct a study in Argentina. I think Daniel, he went to Argentina, and Dr. Angela, and they use the goat. The facility is easier than US. You know, US is that complicated and restrict to BL3, especially when they are with hot strain. So I think they are conducting more studies. And you know, like Lauren, she used like an inhalation. Martha also, you know.
I mean, it took them several years to develop it. So it's not easy.
I guess.
So I don't think that it's easy to give up on a promising vaccine. So you have to do several studies in order to conclude in the end if it's protective or not.
Exactly.
And whether it works on cattle, not working on swine, or I don't know, in sheep, you have to try it. You have to try it to conclude that it's not protective or it's protective, you know?
Yeah, one study I think is not enough, as you said, right?
Yeah. But all of these studies are available either through me or through Dr. Omar, because we were in the same lab, so we can provide all of these interesting papers regarding the Delta VGPR technology. So my due diligence was basically to develop, not to develop, but to do the trial, the safety trial of the Delta VGPR in swine. But other former lab mates did the trial with militensis, I think Canis as well. So all of these papers have been published, and we can share all of them. If anybody is interested, either about the technology of the encapsulation or the Delta VGBR or anything about brucellosis, these remain like very, very good studies.
Exactly.
Yeah, thank you very much, Dr. Slim. But I was expecting more abortion in the S19 group.
Yes,
but we didn't see that.
That's what we were expecting. Honestly, we were expecting, and keep in mind, this is a fairly high dose. So it's 210⁹ CFU.
Yeah.
So we were expecting that S19 group will have abortion, but relying on that. So this is kind of like the second study in swine. The first study was done in 1950s, and as I said, mitigating results we cannot conclude from that paper if the abortion was due to the S19 itself or was due to the infection in the endemic herd. So basically, my study was the unique and first study using the S19 in swine. So and keep in mind, this is not S19, is the Brucella abortus, and it's not the and pigs are not the preferential host of the Brucella abortus. So you can assume that with this high dose you will have abortion, but at the same time you can not neglect that you won't have it. So, and that's what we saw. So basically, we did not see any abortion. So what we are saying is S19 or the S19 delta VGBR, both of them can be safe.
Oh, and this animals, exactly.
Yeah, yeah.
I mean, you did like two in one, you know?
Yeah.
Well, you tested S19 and S19 VGBR.
Yeah.
Swine is a good model as well, you know, if you think it's a
Yeah,
good
yeah.
study.
You used the swine because your target was the wild feral, so it's so that's why you use the swine. And I wonder why. So you did a lot of testing for the humoral immunity, but you didn't mention any about the cellular. So so what's the reason
Yes.
for that?
Actually, so they both are important in terms of protection, cellular immunity and the humoral immunity. But in this study, we basically did not look for the protection, because you can conclude that there's protection after doing the efficacy study, right? So you vaccinate and then you do the challenge. So then you can estimate if there's a protection, either humoral or cellular protection. But for our study, it was mainly to prove, since we have not seen any abortion, we were in doubt that even we vaccinated the animals.
Okay.
So that's why we did that humoral response, and we discovered that the humoral response in the swine model is very transient compared to the cattle. So this is kind of like a specificity of the swine as well. Like whether if this transient humoral response is correlated with a transient cellular immunity, we don't know. So the study is not continuous. We have to do the challenge. But my study was limited to the safety study, so that's why I kept like saying it's an only safety study.
Yeah. It's very great information. Thank you very much for sharing.
And Dr. Sarad, he has did his master with Brucella, I think militensis.
Yeah, I did both militensis and abortus in the guinea pig model.
Oh, that's very interesting. So very interesting. So did you present any? Did you have any presentation? And
no, this was a long time ago in 2008.
Okay.
And we at that time in Iraq we used the whole bacterial antigen. So and we used for Brucella abortus and militensis, and we used a mixture between both bacteria, and we tried to see if there is a cross immunization between the two, and if there is a protection from either. Yeah, which we had at that time with the guinea pig model.
Okay, I see.
Yeah, at that time we had about two abortion in the control group, with no abortion in the just one. I think with Brucella militensis and immunized the group, we had one abortion. Yeah.
Okay.
But the rest were clear.
Okay.
Yeah, but there were bacteria inside the animal. So with the isolation, we isolated the bacteria from the animal, but it were protective against the abortion.
Okay, but basically you did kind of like bacterial study, and you were able to isolate bacteria basically from different tissues.
Yes, at day 60.
Okay.
Yeah. We isolated from different tissues, and we compared between the groups, and also with pathological study. Yeah.
Like here, I don't know about the endemicity in Iraq or also because I know in Tunisia, Brucella is very endemic, and here in Canada or in the US, it's mainly limited to the wild animals.
Yeah, in Canada and US, that's the case. I was hoping that some of the clinicians in the University of Baghdad being present in the workshop, so they will comment on this. But I think Iraq is endemic with Brucella, and we have used depending on my information, I'm not a clinician in the field, but it's I think we are using S19. We used limitedly RB51 in the field, I think, and but it's still Iraq endemic, and we have a lot of cases. But recently there is no new survey about that.
Yeah, and even in Tunisia, I remember there are also like human cases through unpasteurized milk, stuff like that. Yeah, and also
same thing here.
And also cheese, raw cheese. People eat raw cheese in Tunisia, unpasteurized raw cheese. So
yeah, because it's more delicious. But that's not safe.
That's what they say, but they don't know.
Yes. We have the same in Iraq. We don't. It's not commonly the cheese. It's what you got, the cream.
Gamer. Hey, gamer.
They say the cream is pasteurized because when they made it, it's under heat. But it's the heating. It's not that efficient to kill the bacteria.
Yeah. So other than that, basically right now I'm doing my PhD and I switch gears. So I'm not working anymore with Brucella. I'm working right now with mycobacteria. It's more dangerous.
So you mean mycobacterial TB?
Yeah, TB.
Okay, yeah.
Yeah. So I unfortunately, I cannot disclose so much. Most of my work is vaccine development. So as I said, I'm part of the vaccinology program. So most of my work is vaccine development. But on a nutshell, I would say that I'm modifying a BCG vaccine. And there are different BCG strains like in the world. So it's not only one strain. So there are multiple strains, over 13 strains existing in the world, and I was able to modify all 13. So this I won't do the efficacy study. Maybe someone else, but most of my research is involving the development of vaccines, either through BCG or another species, modifying another mycobacterial species.
So if you could comment why, so TB is also, it's not that common in Canada and US. So why there is a focus on the TB vaccine?
Basically, you know that Canada has, honestly, so let's talk about Canada here, not about the US. So Canada is very endemic in the north population. So the Aboriginal population up north, exactly in Nunavut. Nunavut is very endemic, so it's very comparable to Africa's situation.
Oh,
that's real,
and they don't vaccinate. So Canada, they don't vaccinate against TB. So these people, they never encountered any protection against mycobacteria. And at the same time, remember, Canada has a huge immigration policy, and they recruit lots of immigrants, and people tend to immigrate, and most of the people, they come either from endemic countries, and at the same time, remember, refugees from Eastern Europe. So Eastern Europe is very endemic in TB, and at the same time, they had so many immigrants, and most of them, they did not do the screening.
I see.
Yeah. So it's a huge burden. It's a huge burden worldwide. Let's not like discriminate and say it's only in the limited resources countries, but it has a huge burden everywhere. And at the same time, my vaccine program is not targeting only humans, but also animals. You see what I mean?
Yeah.
Sure.
So, Dr. Slim, just I don't want to talk about your PhD study, but I've used in vivo and in vitro.
Can you say that again?
Have you used in vivo and vitro, like a model?
So basically, you know that mostly mycobacteria infects the alveolar macrophages. So in vitro, we infect THP1 cells. So these are macrophages. And at the same time, in terms of in vivo, I did not try my BCG vaccine, but I tried the different species that we modified. And it's comparable to BCG. It's very comparable to BCG in terms of protection. And at the same time, we can discriminate between basically vaccinated and infected through the PPD test. So they are not reactive. Our vaccine is not reactive to PPD test, whereas the BCG vaccine is reactive to PPD test. You see what I mean? So you cannot distinguish vaccinated from with BCG from infected people with mycobacteria. Yeah.
Oh, nice. I guess it's a huge work,
A
right?
huge. It's a PhD. It's a PhD level PhD level study.
And it's nice in the University of Saskatchewan. I know you have a good facility for BSL2 and maybe BSL3 animal.
Yeah. So actually, I'm part of Vido Intervac. So Vido Intervac has a history in vaccine development, and also basically not only history, but also they have the facility. So they have the level 2, they have the level 3, and very soon they will have the only academic level 4.
Oh, nice.
Level 4 in Canada. Yeah,
and it's for large animal, correct?
It's for large animal, correct? Yeah.
They have the same policy as US, Dr. Slim, for BL3? Enroll in this programming.
No, so for me, I have to get trained. So you get trained, and then you go like whenever you have work in level 3. So yeah, and cameras everywhere. You know how you swipe.
But the system is still there or virtual?
You can work by yourself, but you have to let know the security that you're alone, and you have to call the security, I think, every hour.
Okay.
Yeah.
You remember, like in US, first we started with the real body. So friend or your colleague, he has to come with you inside. And they switch the virtual system. It's be easier, and because I can stay with you for how long? It's not easy to stay inside waiting my friend inside the lab.
Yeah.
It's good. I think even in BSL2 labs or animal facility, they have required the body because it's important for safety.
You never know what happened, but here they have another strategy which is not all of us, we get trained, and you can go by yourself. You only need to declare that you are entering and you are exiting the level 3, and that's it. And you're by yourself. You have to let them know by yourself. If you go with a buddy, you don't have to call the security. You only call the security when you're only by yourself.
And you have to sign and everything?
Yeah. For entry and exit, yeah, of course. Entrance and exit, you have to sign all the time. And we have also like a facial recognition system, all those fancy things, when you enter. Yeah.
So yeah, it's more like a development than US. I mean, that's it.
I mean, you remember, Omar, the level 3 facility in Texas A&M was not built, but they basically copied the one here in Vido.
Okay.
You know that there was kind of like a back and forth from Texas A&M to University of Saskatchewan just to get the expertise and know how it's built and everything. So the one that was built recently at Texas A&M and is evolving right now at Texas A&M is kind of like the copy of Vido here in Canada.
Nice.
Yeah.
So they mean that you mean the BL3 for large animal?
Large animals, yeah. The one that was recently active at Texas A&M.
Exactly. Yeah.
Yeah,
I think Dr. Sankar and Dr. Allison faked she is in charge.
yeah, and Melissa.
Oh, Melissa, yeah.
Yeah,
Oh,
Melissa. Yeah, and we received the director of the facility, I think two months ago here at Vido. He gave a presentation and he mentioned that basically.
interesting.
Yeah. I mean, you can look it up, like Vido Intervac here at the University of Saskatchewan is very famous for vaccine development, vaccine trial for animals. Large animals can be also small animals because the facility can hold small animals as well. And yeah, we have different programs, different pathogens. We have TB, we have influenza, coronavirus. So people, they work on llama, alpaca, different species, cattle, many species.
Yeah,
Thank
great.
you.
We might be sometimes need some some presentation about such facility, because it may be you or someone else from the facility to just show people here in Iraq how they what things available in other countries, what facility, how it's the resources, how it's get managed, because it's a huge thing to be or the BSL3, because I worked only in BSL2, large animal facility, which is huge, and so I think BSL3 will be very beneficial and good to know. I don't think one time we're gonna have BSL3 in Iraq, but it's nice to see.
Yeah.
No?
So you don't have any BSL3 in Iraq?
I'm not sure. Let's Omar.
I think we don't have this for my
Not even for small animals like mice, rabbits.
I don't think so. No.
No, we don't have.
I don't think we have BSL3, but we do have BSL2 work in BSL1 lab, but it's because Iraq is more endemic in many things, so it's not yeah, even my work with the Brucella, we did it in BSL1 lab. I don't think.
Okay. Yeah. I mean, in Tunisia, they through Institut Pasteur, so Pasteur Institute, they have recently inaugurated a new level 3, I think 2 years ago, but that's only for mice work.
Yeah. No, I don't think we have in Iraq.
In Tunisia, yeah.
Afan, Dr. Sarha, just the link, some of the.
I already sent it several times, but I'm gonna send it again.
I don't know, it's not working, they said.
It should be working. Can you test it with from your side?
Okay, just a second.
It's work with me.
It's working, yeah, exactly. الاسم معلقة مكان العمل, it's clear. So
and I have 94% signed up.
لسادة الكرام الرابط ما بي أي مشكلة إن شاء الله. أخاف مشكلة بالإنترنت يمكن مثلا.
If there is any question, please raise your hand and we will unmute you and let us know. Otherwise, Dr. Amar.
I see Dr. Amar who said hi. I couldn't. I just saw his message. Yes, hi Dr. Amar.
He's greeting you. Yeah.
Former colleagues at Texas A&M University.
Any question, Dr. Salim? Salim or Slim, Doctor? What we have to call you? Slim, right? It's not familiar. I mean, this.
We had
Salim.
We had this conversation with another colleague from Egypt. His name Tamer, and I always call him Tamer.
Tamer. Yeah.
He said it's Tamer, very different from Tamer. So I thought they are the same. So then he explained it's a completely different name, different meaning.
Other than that, Omar, I think, I don't know if there are students interested about the Fulbright program. I looked it up like very quickly, so I don't know, you can correct me. So there's, it's through Amidist, and Amidist is in Erbil.
Arbil, okay, محافظة.
Yeah, yeah. So basically, there's the Fulbright program for masters exists in Iraq. So if there are any students after completing their studies are interested in doing masters in the US through the Fulbright program, they can look it up, and it's called Amid East. Amid East.
Can you share the link, please? If you open the link, please.
Amid East, and I will open the link. Yeah,
and I think it's also available through the US Embassy website.
Yes. Yeah. So usually they display that the US Embassy. Sorry, they usually display that and talk about it.
Yeah.
So it's usually for masters, and you will get like your finance entirely, like to do your masters in the US. It's a lengthy procedure to apply, lengthy application, but if you feel like you are a distinguished student and you can have an input, you can apply for it. And I mean, good luck for everyone. Every country in the world, so it's not only limited to North Africa or to Asia or to as I said, limited resources country. You can find people from Australia having the Fulbright scholarship, people from Sweden, people from Germany, everywhere in the world, or even in France. So, I've met many people through presentations and through the program, through the Fulbright program, from many countries, like from Iraq, from I remember from Afghanistan, from different countries, you know.
And I think your boss was in Washington D.C. The guy.
Yeah, so I used to have an advisor, the Fulbright advisor. So yeah, he was in Washington D.C. Yeah.
Interesting.
Yeah, and they invite us for seminar. As you remember, Omar, I think I went to Washington D.C. I went to for conferences. So all of these are kind of like Fulbright conferences, and they are kind of like interest different categories, different fields. So for me, it was in the health field, basically. So we had our seminar related to the health field, like public health, master in biomedical sciences, all of these branches, you know. So we had seminars in Washington D.C., but also at Kansas, I remember. Yeah, so this is a good opportunity for people to study in the US for free.
This is the main thing, exactly, for free.
For free. Yeah, for free.
Yeah.
At no cost.
Thank you very much, Dr. Slim, for sharing.
Thank you so much.
Any time, and if anything is needed, as I read through the different messages, encapsulation procedures, I can share papers either through Dr. Omar, or you can contact me through my LinkedIn. So my name is Slim Zriba. I have my LinkedIn always. I can share anything through my LinkedIn, and we can share information. Just reach out. Feel free to reach out at any moment, and Omar can leave my my email as well.
Yeah, sure, Dr. Omar. You can have this information posted on the website when you write the summary for this workshop, and you could post the link for that paper in
Facebook.
It's about sharing knowledge. Everything is about sharing knowledge, and to improve our knowledge, whatever it is, either in Canada, US, Iraq, Tunisia, whatever in the world. It's about sharing.
Yeah, that's the objective of this, and thank you for sharing your knowledge with us.
Yeah. Thank you very much for the invitation, and You're welcome.
Thank you so much for your time. I know
Yep.
it's hard, I think as you said, minus 19 or 36, and you travel to the video. Thank you
Yeah,
so
yeah,
much.
yeah. I have some work actually, so I have to work. Yeah, so it's not a problem.
And we have to meet you again, inshallah, after you finish your journey. Okay?
Yeah,
And
yeah, for sure.
program, a PhD.
Yeah,
And-
yeah, yeah. When everything is possible to disclose, and if there are any publications or whatsoever, I can talk about it.
Of course, sure.
Yeah,
Thank
yeah.
you so much.
And sorry again if I was talking very fast. I'm sorry about that again.
Thank God you didn't mix French with Arabic and English, and it's gonna be super hard for us, you know.
Yeah, yeah, yeah.
But I know how you speak like a Tunisian now, right? I learned a little bit from you.
A little bit.
A little bit, exactly. Thank you so much, Dr. Slim.
Yeah, you're welcome.
Bye-bye. Thanks.
All right, thank you guys,
Thank you very much.
Yeah. and see you.
See you. Bye-bye. Take care.
Bye.